Thank you very much for the invitation to speak today. It's a great honour to speak to everybody. What I'm going to talk to you all about today is in the title, the genetic methods for the control of honeybee parasites and pathogens. But we are going to start off a little bit on wasps and conclude a little bit along the lines of, well, what is the future and how do we progress this area of research? I'll start off, so I'm an entomologist. I've been here at Victoria University for the last 20 years or so. We work on social insects. So primarily we've done a lot of work on things like Argentine ants. These are some Argentine ants in a beehive here. We've worked quite a bit on wasp populations and also on bees and their parasites, like this little one here, the Varroa mite. Right now, we are very reliant on chemicals for the control of these, for all of these pests, right? So Vespex is just a wonderful baiting system for the use of and controlling wasp populations, right? So this is a New Zealand invention. Richard Toft down in Nelson created this, and it's hugely useful. It contains fipronil, the pesticide. For Argentine ants, the only real option in New Zealand, again, is the sort of fipronil-based bait systems that are really effective. This is Vanquish Pro ant bait. It's, again, a fipronil-based thing. We've got, this was a Varroa strip here coming into a beehive for the control of Varroa mites. These are really the tools that are a mainstay of pest control in New Zealand. What we are doing really is working with the biological heritage program of the New Zealand National Science Challenge to do things like this, to develop socially acceptable, cost-effective and highly targeted next generation technologies with the aim of mitigating the impact of invertebrate pests in both natural and production ecosystems. What we want to do really is move away from pesticides, right? So how do we get away from these what are really broad spectrum pesticides that can have an awful lot of non-target effects on populations, on communities out there? What are the next generation? What are the highly specific, highly targeted tools that we can utilize to control invertebrate pests? And we've been looking at a bunch of genetic methods for those. I'm going to have today, we're going to talk about three things, right? So I'm going to work through really some novel technologies, novel genetic technologies for the use of controlling invertebrate pests. I'm going to do a little bit to start off with in terms of a review of our work on social wasp populations, right? So these are a massive issue that I think really needs something and something really soon, if possible. But then I'm going to move on to another pest in social insects as Varroa. Varroa is parasites of bees, and work through a bunch of those so that we've been thinking about. So utilizing double-stranded RNA for Varroa control, using genetically modified bacteria to secrete that double-stranded RNA, or a little bit on Varroa and gene drives in Varroa. So there's some work that's developing in this area that could be of use down the track. What I do think is increasingly obvious out there, and I'll finish up with this, is that we are starting to really need a loud, proactive, balanced voice to encourage scientific and legislative research in this area. So I'm not saying here, and I go to great pains to not say that we need to start using genetically modified organisms or releasing them in the environment right now, but I think we need to really start considering the utilization of these sorts of technologies and what that means to really balance out the voices at the moment that are against this technology, right? So we need to start thinking about how we do this, how we promote the need for research into these. And I'll finish up by talking about that. But I'm going to start talking about genetic work on social wasps, right? So what have we been doing on social wasps? One of these are one of the key problems in New Zealand's native forest. What I wish I could do really is take the Minister of Conservation and lots of high power people into the forest, into our honeydew beech forests in April, in January, February, April of every year, and just put them there with a jam sandwich and a can of Coke in their hands to see what happens and to see the abundance of wasps in these populations. Right, so if we look at vespular wasps, primarily Vespula vulgaris, the common wasp, if we look at populations down there, then the biomass for those is as great or greater than the combined biomass of birds, rodents, and stoats. Right, huge populations down there. Massive effects, right? Up to 10,000 workers per hectare, 370 workers per meter square of tree, over wherever there's really honeydew beech forest, and they change it substantially, right? So this is a tree over the side here, and you can see lots of little honeydew droplets all over this tree. You walk through the forest, and you can smell the air is sweet from all the honeydew. It's a natural thing that's going on in those forests. If you go now into lots of those forests where there's no wasp control, you see a huge abundance of wasps everywhere that really change this. You'd hear a drone, you no longer smell the smell of honeydew. It's a very much a changed ecosystem and down there now. What have we been doing? Well, one of the first things we did is go down there and look for sources of naturally occurring genetic population variation that may be associated with reduced fitness. So this is some work led by a PhD student, Jana Dobelman, with Neil Gemmell and some European collaborators and myself. We've been looking at mitochondrial genome for naturally population indicators of genetic variation that are associated with reduced fitness. So if we look here, we've got haplotype over the side here. We've got 4 different haplotypes, mitochondrial variants going on here, and number of workers per nest in those populations. And you can see, okay, there's around an average of 5,000 to 10,000 workers per nest in these populations. And for the most part, they produce a whole bunch of queen cells, right? So about 500, 250 to 500 queens every year. But there's the rare populations out here that are associated with reduced fitness. They don't produce a whole lot of queens in those populations. So yes, there are some mitochondrial haplotypes out there that are associated with reduced fitness. They could be perhaps a suitable system for a Trojan female control system that Neil has done a lot of work with and talked about quite a bit about. But equally, I can't really see the New Zealand government or the New Zealand public buying into mass producing of this variant every year and the release of these queens throughout lots of systems in order to enact a Trojan female system in wasp populations. And I think that's pretty fair. We've also been looking at CRISPR gene drives and doing some preliminary work around those with Peter's group down here and John Keane from Ag Research involved there as well. So this is doing things like, well, okay, what could be the target genes that we'd look at? And so these are a series of 1, 2, 3, 4, 5, 6, 7 different genes that are involved in the reproductive biology of wasps, and looking at the variation associated with those here in the natural range, right? So if you increase the number of your samples of wasps, then you see an increasing level of nucleotide substitutions or genetic variation in wasp populations, different levels of that with different genes. People are concerned about that level of variation, especially high levels of variation in terms of resistance development for gene drives out there if we were to use them. And what we've found is, okay, there is variation out there. You can select for genes involved in reproduction that have almost no variation, or genes that have a lot of variation, and utilize those. So we're trying to get a handle on, well, could resistance develop out there? Well, it might be able to develop it if you utilize genes like this one, but if you use genes like this one, then there are very little genetic variation and resistance probably wouldn't develop out there. So there's limited amounts of genetic variation in the New Zealand range. It depends on what genes you target. We could target genotypes in the New Zealand range that are distinct and are specific to here and leave genotypes in the native range, other genotypes, unaffected. So there's potential there, I think, for developing a highly specific, highly targeted gene drive that would attack and only effectively affect the genotypes in New Zealand, plus some of the genotypes in Europe, because it's where they came from, but not all of the genotypes. So what we're trying to do with a lot of this sort of research is to start thinking about, well, could resistance develop? Could we target very specific genes that would only affect genotypes within New Zealand and not all genotypes everywhere? And the answer is yes, we can look at genotypes or target genotypes quite specifically within the country. We've been doing some population modeling, right, and some work looking at spatial variation and how spatial variation affects populations throughout the country, right? So this is clearly a map of the South Island. This is the purple bits are where this honeydew beech forest, and highly, dark purple is high densities of wasps and honeydew beech forest. Light purple or nothing is very low populations of wasps. That's this is a simulation model, which I hope runs okay over Zoom. And over the side here, this is a graph showing the overall densities over time. So I'll run this, and you'll sort of see this populations will fluctuate. And the colors will change here a little bit as we progress. And so I hope that comes across over Zoom okay. One of the things you can hopefully see on that is, okay, we do see populations being reduced if we implemented a spatial gene drive in these populations, and if it was successful and if it did establish. What it seems unlikely from these simulations, at least, is that we would end up with eradication. What's much more likely is that populations would be lowered. How much depends on the gene drive, quite substantially. And there'd be sort of outbreaks in little populations at little times and all that sort of thing, while there would be lots of areas that would be experience low densities of wasps at different times. One of the things you will notice on this is that this scale is in years, right? So it would take 20 or 30 years for us to experience a population crash overall, but then populations would cycle after that. So some interesting results, right? So there is potential, what it seems here, to lower wasp populations via a gene drive. It could take populations down, but what seems to be the case is that we would have persistence overall of wasp populations over time, right? So we wouldn't see eradications, we'd see populations persisting over time, not getting rid of them entirely. That's kind of all I wanted to talk a little bit about wasps before I moved on into honeybees. But look, I would say that we need to start thinking about genetic modification of wasp populations via genetic means. It could be a self-sustaining, it could be a cheap solution to what we are experiencing here in New Zealand is the highest known recorded densities of wasps, right? In large areas of our beech forest. This is a huge problem that we need to really examine and to utilize some sort of novel pest control technology within. It would, however, take decades to achieve, right? I suspect, given the dynamics we've seen in our spatial modeling, that would probably be a case of reduced densities rather than eradication, though there could be ways and means of getting around that. I think we really need research into the efficacy and impacts of gene drive technologies and genetic modifications in these systems, right? So there's things like biological control that might be an option. Could have some impacts. There are genetic methods probably offer one of the very few alternatives over that 1 million hectares of area that they're infected, right? We do need to worry about the concern of these wasps being transported back to their native range. So we need to think about mitigating that, right? Because in the native range, there are people that for whatever reason, really love their wasps, right? So Syrian Sumner is one of these, one of the people who really promote wasps in their native range, and fair enough that she loves them, and you should too, apparently. And they are, at least in the native range, considered hated but misunderstood. You know, they do have a role in pollination. They do have a role in pest control in the native range that probably more so there than here, where we don't like them quite so much. All right, switch gears a little bit. What I'm going to do now is really start talking about Varroa as parasites of bees, and what we really need is some new tools for their control, right? And I think genetic techniques offer some real opportunities in here to be able to do that sort of thing. So I'll start off with a bit of a description about Varroa. Lots of you will be familiar with Varroa. Some of you won't be. This is the Varroa parasite over here, right? So that's on a bee pupa, right? So these are two adult female mites. They're parasites. They feed on the fat bodies of the developing pupa, right? But one of the things they also do is they transmit something called the deformed wing virus. All right, and there's a bee in the background over here with normal wings. This is a bee clearly with not normal wings, right? This has got deformed wings associated with, and she is sick. She's got a little less hairs on her. She can't fly, clearly. She'll be kicked out of the hive in due course and die outside of the hive. She's useless to the hive, does very little for it. This is a bit of the life cycle of Varroa and some more pictures of it. Here's the Varroa on the adult bee. This is a side picture of it, right? So they will often, you're hard to spot necessarily in your hive. They will hide, this is a honeybee, they're hiding in under here, where they are sort of snuggled in up here. Within side here, though, they are feeding on that bee, right? So the little mouth parts are penetrating the bee, and here, close up of those, where they are feeding on that adult bee. They are potentially transmitting viruses at that stage. What they'll do is the queen life cycle was such in a hive, the queen will lay an egg. Here she is laying an egg. That egg then develops, carries on, that's fed by a worker, a nurse bee comes along here. When that larvae gets to a certain stage or age, the mite will crawl off from that worker bee, the nurse bee, go down, hide in amongst the food down the bottom here, wait till the cell is capped, and then it'll start feeding on the pupae and reproducing on the pupae, develop and then leave on the new bee that develops from there. Only a couple of weeks in terms of the life cycle. So actually that's quite a bit faster than the wasp that we're talking about, which has an annual life cycle. We'll get to that in a bit. Varroa are a huge problem, right? So lots of you again will be familiar with this sort of information, right? So this is estimated colony loss rates within New Zealand. This is some data from a couple of years ago. This is from 2018, New Zealand Colony Loss Survey. MPI are involved in this, Landcare are involved in this, and you can find lots more information about it from this website. We have relatively low rates of colony loss within New Zealand, so we're looking at around about 10%. It seems to be increasing though over the last few years. What are the reasons for that? Well, queen problems are a common reason for that, and likely some of that is due to just queens get old and die, right? So they get old and die. But amongst lots of the factors that we can do things about, Varroa are a huge concern about those. 20% or so of hives are lost, are lost because of Varroa. Wasps are a big problem. Argentine ants are a big problem further up north that you go as well. So Varroa are a critical problem for our beekeepers in New Zealand. We need to be concerned about those, right? Beekeepers I know will commercial beekeepers will spend $50,000-$60,000 a year on trying to control Varroa in their hives. So it's not cheap. It's not just Varroa, as I say, it's with the deformed wing virus, right? So Varroa's risen out here in Indonesia and Asia. It's moved across into Europe and then throughout the world. Wherever it's gone, it's taken the deformed wing virus with it. Right, so it's spreading that around the world, and certainly the global honeybee viral landscape has been dramatically altered by this parasitic mite and the virus that it transmits. It's a global epidemic of this virus cause by the distribution of Varroa in the systems. So it's sort of a hand-in-hand thing, right? So we've got Varroa and we've got deforming virus coming along and being a major problem now around the world. It is a major problem, right? So this is a Trends in Parasitology piece of work in 2020. Varroa destructor is an underestimated parasite. I don't think, there's lots of people out here that would disagree with that. Lots of people that would say Varroa is the greatest threat to honeybee health, and we know that already. But it is getting worse, it seems, almost. Worrying observations include increasing acaricide resistance, shrinking economic thresholds, treatment thresholds, and the mites affected are becoming more, and the viruses are becoming more virulent. If virulence is not punished, it will proliferate. I love that quote from the study. And it's saying that really what we're doing via our treatment of Varroa these parasites, by saving hives, by not letting them die and all those sorts of things, is we're only selecting for more virulent genotypes within the hives and within Varroa. Right, so they're predicting here this problem is only going to get worse in the next bunch of years. Right, so we've got some issues that are coming up. And last little study, this is a bee from my lab, right? You can sort of see a little Varroa mite under here. This bee has generously lifted a leg to show us all this little Varroa mite that's hanging out underneath there. This is a study from 10 years ago, and it says, look, we've just not achieved the original aim to get rid of the parasite, or at least solve the problems associated with Varrosis, right? The diseases it carries. There is neither a Varroa treatment that's available that fulfills this criteria of safe, effective, and easy to apply, nor a honeybee which is tolerant to Varrosis in the Varroa under temperate climatic conditions. I don't think we've moved on from that, right? We're still in that phase where we've got these problems that are associated with Varroa. We need some sort of solution, and the solutions that are available to us in terms of parasites, in terms of chemical controls and pesticides, are becoming less effective out there. Using double-stranded RNA, right? So I'll talk about using double-stranded RNA for Varroa control, and something we've been doing a little bit in our lab. This work really stems from a paper in 2012. We're looking at double-stranded RNA for or gene silencing for Varroa control. Right? So lots of you will be familiar with RNAi, double-stranded RNA, using for pest control out there. Basically, we've got a system where they've got a nucleus. The nucleus has got some genes. You want to have a gene expressed. You produce some messenger RNA. You send that out of the nucleus. It goes to the Golgi apparatus to get produced into a protein. When you want to stop that, you use can use an RNA interference way. You've got that going on in your cells right now, where you've got short interfering RNAs produced that really dices up effectively in the end, degrades the RNA so no protein production is being produced. This team utilized a way of doing that. Starting off, okay, they had some RNA, double-stranded RNA that they fed to the mites. You can feed it or inject it or do the feed to the bees. And as a consequence, there's RNA, double-stranded RNA from the outside, sort of referring to it here, stops that production of RNA, stops the gene protein production in the mite, and can kill the mite. Right. So this work quite a while ago now, right, almost 10 years, looking at okay, they've got a mixture of double-stranded RNA, 14 different genes that they were targeting in the end, and they were able to reduce Varroa populations by an average of 53%, all right, and 61% compared to an untreated control. So there's positive things there, able to reduce using a highly specific, highly targeted way, introducing some synthesized double-stranded RNA. They can stop protein production in the mites and effectively kill the mites in these populations, right? A whole bunch of genes that they're targeting. This is not especially important at the moment, but here's a list of the 14 different genes that they were targeting in this situation. There has been other work since that time that's looked at a bunch of different genes, Right, so so here's a study where rather than feeding honeybees, they're directly injecting little varroa mites. This is a direct injection, and post-injection, they're seeing mite survival crash dramatically as a consequence, right, and compared to untreated controls. Right? So it's possible using RNAi in a bunch of different studies to be able to knock down varroa populations as the end story. Some of you will be thinking, right, Bennett, well, that's nice in the lab, can you upscale, can you apply that to field situations? And the answer is, well, actually, we're starting to see studies and companies do that sort of stuff, right? So this is from Greenlight Biosciences, and we've started a relationship and some work with this company in the US who have a goal of producing sustainable solutions to protect the health of all people and feed our planet. They've got a program on honeybees, utilizing the double-stranded RNA sequences from the Israeli group, and some work in 2000, as far as 2016. They were using field trials, so putting double-stranded RNA in honeybee hives in the field of 2,500 colonies, right? So they've got a production platform that's able to utilize this technology, produce double-stranded RNA in a big way, right? Enough to be able to get to a commercialization point just about in terms of varroa control using these techniques. So we are quite a ways down the line here of getting a next generation tool potentially for varroa control in honeybees. I think we should really be starting to think about this as a technology that could be applied in New Zealand, and we're investigating that. Going on from that, right, the next sort of step or level from that is utilizing genetically modified bacteria to secrete those sorts of RNA sequences that were being utilized by those Israeli groups. And this was a study last year that was looking at, published by Sean Leonard and Nancy Moran's group in Texas, right, where they're engineering similar, so they're genetically modifying gut bacteria to activate honeybee immunity and limit pathogens, right? So what they have done is come along and genetically modified some bacteria that to secrete double-stranded RNA in the gut of some honeybees, right? And here's a picture of that. They've fluorescently labeled it here, right? They're able to do that. They're able to inoculate the honeybees, be able them to have the bacteria culture inside the honeybees, be able to secrete that double-stranded RNA, and be able to affect mites and pathogens associated then. And Science had this on the cover of their magazine for that issue, and really was starting to think, hey, maybe this is a microbiome silver bullet for honeybees. Maybe beekeepers could get to a point where we can feed our honeybees these bacteria, the honeybees that have the bacteria established within them, is secreting the double-stranded RNA, and it's controlling the parasite. Really kind of potentially a really cheap and effective way of going about doing this, right? So these people think, well, is this a silver bullet? Is this a cure for really varroa populations? Lots of work to do there. I think there's good reason to be excited about this, right? But there's still lots of work to go on here. What they are finding here is, okay, the symbiont-produced RNAi, so gene silencing, can improve honeybee survival after viral injection, right? So there's not enough time really to go through these in great detail, but they're injecting bees with deformed wing virus. They're looking at the survival of the bees. And certainly there are significant effects that they can significantly enhance bee survival after injection with virus loads as a consequence of these gut bacteria. And the same with Varroa, their work on Varroa, right? So symbiont-produced RNAi, so double-stranded RNA produced by these bacteria, effectively seems to kill some Varroa mites feeding on the honeybees, right? So lots of work to do here still on this region. We're not at a silver bullet yet, but I do think that there is opportunity here that this could be an effective solution for Varroa control in honeybee populations. I'll finish up by just, in this section at least, by talking a little bit about gene drives. And people certainly are starting to think about gene drives for Varroa control populations, right? Not necessarily effectively, unfortunately. So here's a gene drive. This is a preprint that's just come out on a preprint server, right? By a group was looking at a gene drive for Varroa destruction, and unfortunately found, well, okay, gene drives don't spread easily in populations of the honeybees, parasite Varroa destructors. So people are starting to think, well, we could utilize some gene drive technologies for Varroa control. And what they have found as well, look, a gene drive that's really targeting female or male fertility in Varroa doesn't spread easily. And that largely reflects a bit of what we've found here in looking at wasp populations too. Look, the harder the gene drive is on wasp fecundity, the slower it spreads through populations, right? But I don't know, that maybe we can think laterally on a bunch of this and start thinking of, well, rather than affecting fertility, could we use it to suppress popular levels or amounts of deformed wing virus in these populations? Could we associate it with some of those genes that we've looked at before? What would happen in those systems if we incorporated suppression of deformed wing virus in Varroa populations rather than looking at fertility of the mite directly? I think that's worthwhile sort of thinking about and doing some modeling on, at least. To sum up that sort of situation, right, the double-strand RNA delivery or genetic modification of bees, or perhaps even the suppression of viruses within mites via a gene drive sort of technology, could really be a self-sustaining, could be cheap, could be novel solution to what is the world's, not just New Zealand's, but the world's worst honeybee problem. Right, so people all over the globe are looking for solutions to this problem. Right, it's a major issue. It's, I suspect, much faster, certainly with gene drive technologies, because the Varroa have a population cycle lasting just a few weeks rather than a year for wasp populations, for example. It could be much faster action in wasp for Varroa, right? Gene drives that result in a reduced Varroa densities are good, right? And if we had low populations of Varroa in a hive that were lower than economic thresholds that are out there, well, that's actually a good thing, right? To maintain those in there, because you've still got a mite representatives that can keep the gene drive going, keep lowering the population of Varroa within a hive. Whether or not that could happen is an interesting thing. We've got lots of modeling to do on those sorts of situations. We do need lots of research into the efficacy and impacts, both social and ecological, in this area. Right? That's clearly what's needed. And I'm, for one, am not saying we need to implement double-stranded RNA in our beehives tomorrow or the next day, but I certainly would urge us to really consider this as a viable control option, but we need more research in there before we can make the conclusion, hey, we need to start doing this. I would say that, look, everybody hates Varroa. There's nobody in England, for example, that says I love Varroa, or nobody in Asia that really loves Varroa. And we're not talking about the global elimination of these populations either. If we had a gene drive to suppress deformed wing virus, that would probably mean there's just lower the populations rather than elimination of a species, even a horrible parasite that nobody likes from the world. I'll finish up right soon by talking about this final point. I really do think that we need more scientific and legislative research into these novel control technologies. What I'm not suggesting that we do is release any of these things tomorrow, right? But I think we need to somehow facilitate people to thinking about these, and I think we need a loud, proactive voice. I do think at the moment the voices against any sort of technology, whether it's double-stranded RNA or genetic modification, those voices are really louder than those promoting the need for research, and I'll give you some examples of those. All right. All around the world, right now we are seeing the implementation of gene-drive genetic modification for pest control. This is some propaganda, promotion from Oxitec, at least, that have been working around the globe, and right now are doing some releases in Florida populations, right? So they're releasing some mosquitoes, some genetic modified mosquitoes, the gene-drive populations of mosquitoes in Florida, in the hope to reduce human disease in there. But those have been done all around the world, right? So the mosquito work is a really high priority for Oxitec, and they've been doing them in Malaysia and India and Europe, right? So in all sorts of places, but they've also been doing other pest populations as well in North America for quite some time now. They've been looking at diamondback moth control and pink bollworm field releases over there. Right, in Australia, they've been doing, at least in cage releases, some medfly, fruit fly population work, right? So things are happening all around the globe, right? And I think this is a really important one. The fall armyworm is becoming an increasingly problematic pest that we really need to start thinking about. Well, how do we control, especially in a lot of countries where this is a pest that's a problem for people that can't afford any other control technologies or techniques. Right? So all around the world, we're seeing genetic approaches being utilized. There's quite a recent little bit of publicity, and what I'll do is I'll get you not to focus on the maths AU groom tears up during genital cupping exercises, which makes you cry in so many different ways. But this is a feature on our New Zealand TV. Focus instead on Graham LeGros here, who was talking earlier this month about archaic genetic modification laws. And in terms of his point of view, was really stifling progress in New Zealand's COVID vaccine development. And his comment was, well, look, where our genetic modification laws have really stifled COVID-19 vaccine work. Right? And he's saying, look, these laws were implemented 1998, so 20 years ago, and haven't really been modified since, and is not really helping us a whole lot of this, especially in terms of their work for vaccine development. Range of views on that, of course. So not everybody thinks like Graham or the rest of us, right? So Jack Hyman believes this law is still fit for purpose, right? And look, everybody's welcome and has their own perspective, but I do think that the vast majority of us would probably be in agreement with Graham here and saying we need to reconsider these laws. We need to consider what we're doing out there, right? And do things about it. Environment Minister David Parker refused to be interviewed on the topic. He ducks for cover, and I think that's very typical for our minister, unfortunately, at the moment. Right, he's ducking for cover. He's saying, "Look, these regulations aren't part of our manifesto to look at in the future." There's been certainly some work that has promoted, that we think about this. So the Royal Society have done some work looking, and quite recently, looking at, well, do we need to modify these laws and regulations, right? And they could have come out with a harder conclusion, but they considered that New Zealand's statutory provisions and regulations around this need to account for increasingly nuanced view, right? So we need to start thinking about them at least. That was from the Royal Society. I think, again, the minister ducked for cover on this. The minister really has not made any changes or taken into account anything around this. And in the meantime, in the background of this, there's quite a bit of pressure being applied from organisations such as the Sustainability Council on the minister and on the EPA and on a lot of different authorities to say, well, should we think about what is the standard on things like double-stranded RNA in terms of is it a genetic modification? What's going on here? And I believe that they are, and a lot of different people are putting pressure on the minister to do things, but the other side of the coin, where the voices to say we should be investigating this technology, we should be thinking about it. Well, the Royal Society have done some work on this, but it's really been sidelined by the Minister. He hasn't taken a lot of notice on it. I think the opportunity is out there for a bunch of organizations to really start thinking, hey, we need to get together and start thinking in terms of partitioning or talking to the Minister about making the legislative change or thinking about legislative change a high priority. I really would like to see some prestigious, esteemed organization not thinking about anybody, but maybe Genomics, Aotearoa, you know, in an organization which one day I hope to be invited to be a member of. But I realize that these are just dreams and pipe dreams and unrealized goals. Well, maybe these sorts of organizations could take the lead a little bit about putting some pressure on the Minister, getting people together, getting industry together to be able to say, look, we need to start changing or thinking about changing statutory regulations in this area. Perhaps one way of doing it is through the Parliamentary Commissioner for the Environment. Do we as a group of organisations get together and say to the Parliamentary Commission of the Environment, let's start reviewing this situation? I don't know. I don't have all the answers in terms of who we best and how we best effectively investigate and perhaps affect some legislatory change or investigation about legislatory change. It's hard to say on a Friday afternoon, but I think that we need to start investigating that as a possibility. What I am keen on doing is, as I said right at the beginning, is not implementing, not throwing out a gene drive in the population, not utilizing double-stranded RNA tomorrow or the next day for pest control. I do think there's lots of work in here, but I do think it really offers alternatives to these pesticides that we're putting in our hives, the damage that they are doing in their environments, right? And I think we really need to work towards some more socially acceptable, cost-effective and targeted next generation technologies. And I hope we can do that. And I think genetic technologies could be a part of that. All right. I'll leave it there on a Friday afternoon. Thanks to Biological Heritage Program for supporting this sort of research, and a whole bunch of collaborators for being involved, and for Genomics Aotearoa for inviting me along here today. Thank you.